Welcome back, everyone. That is our short break. It has been concluded. We hope you enjoyed that video and the resources that were shared in the chat. And now I'm very excited to introduce Dr. Alba Grifoni. Alba will be presenting SARS-CoV-2 and the ImmunomeBrowser. Thanks, Alba.
Thank you very much, Nina, and thank you for being part of the workshop again. And I always like to bring this up to all the people attending. I used to be one of you, like following one of these workshops. I fell in love with the IEDB, the way they're working, and I joined Alex Lab several years ago because of that. And I'm very excited today to show you the perspective on the other end of an experimentalist that have been using the IEDB. So particularly for this workshop, I'm gonna guide you through how to use the ImmunomeBrowser, and I'm gonna show you some live example. But before doing that, I would like to tease your curiosity to kind of like show you what can you do in your research using ImmunomeBrowser. And basically, I'm gonna try to make example on two recent papers that we've been publishing in this year, applying, of course, SARS-CoV-2 that has been the main focus of our research lately. So talking about these two different presentations, in the first study that we performed, and that was thanks to the help of Alison Tarke, a talented PhD student in our lab, we were interested in addressing the following question. While in the 2020, we were all more focused on understanding what kind of protein the T-cell recognized, now we were more interested in understanding, okay, about what epitope specifically T-cell recognize. And similar to this question, another question that we were curious to understand is, are T-cell gonna be able to recognize the entire protein of spike, or are there any specific immunodominant region that the T-cell specifically recognize? And that was like a question that was very important for us, because in the field, many people were thinking about designing vaccine candidates for SARS-CoV-2, maybe focusing on immunodominant regions specifically for antibody response. So we were curious to see if that would mean that also T-cells were be able to recognize this region or not. And what I'm gonna show you here is the equivalent of ImmunomeBrowser. This has been done with a standalone version of the tool. And what you can see here is the entire spike protein divided for the different domain. The RBD domain is the one, as everybody knows, recognized by the antibody response. And when we look at the CD4 response, it appeared quite evident that the response was not uniform. And actually, there were some specific peacock recognition measuring the N10 domain as well as the CT domain, while the RBD domain was actually poorly recognized for the CD4 T-cell response. So then we were wondering, what about the CD8? And we look at the CD8 response, and that was not the same case with the CD4. Actually, CD8 T-cell response were capable to recognize the entire protein, and their actual response was more related to specificity driven by the different HLA class 1 alleles of the individual we tested. So this type of study was focusing only on a smaller cohort where we have been trying to actually dissect different type of alleles, but then we decided to actually extend this type of analysis using IEDB data. So we performed this meta-analysis in June 2021, where basically what I'm showing you is the same results I showed you before, but with all the IEDB data. And at the time where we performed this meta-analysis, 1,400 different epitopes were available, and they were based on up to 25 published studies. The IEDB has way more data right now, and actually I'm gonna show you what I'm talking about in a few minutes. But essentially, what we found in our meta-analysis was a similar picture. Once again, the RBD domain for the CD4 T-cell response was not as recognized, well, it was indeed recognized for the CD8, and that was true also for other different proteins. Another thing that we noticed in our epitope identification was also that there was a difference in the epitopes. Some epitopes were actually recognized by multiple donors, and actually those account for highest amount of the response. So once again, it was important for us also to capture that, and ImmunomeBrowser allowed you to capture that. And the other thing that we noticed, as I was mentioned to you before, is the fact that in the context of the CD8 T cells, the response was really different depending on the class 1 alleles analyzed. Some allele you have like a lower frequency of response, but stronger magnitude and so forth. And just a comment that I wanted to make to you is the fact that IEDB is actually based on experimental data. So it's as experimentalists that actually feed the database with our own data. And it's important also to point out that the data restriction can be biased, because like if the majority of the science is focused on particular population or in particular most frequent class 1 alleles, then of course, when you're looking at the data set and maybe are interested in looking at different population or more rare alleles, then of course the amount of data changes. So that's something always to take into account. So in conclusion, when looking into the epitope recognized by T-cell response in SARS-CoV-2, CD4 and CD8 have a different way to recognize, and they also recognize specific protein region. And in some cases, that could also depend by the HLA alleles analyzed. And finally, and that was just to reconcile also with the information about the B-cell data, not necessarily immunodominant region for the T cells correspond to B cells and the other way around. So now the question rises, okay, this is what we saw a few months ago. How does the IEDB database look right now? So I am gonna start showing you the IEDB database, and I'm gonna perform this query together with you. So here is the IEDB database, and basically to perform the type of query that we're interested in to kind of mirror the analysis that I show you, we're gonna use the main box searching over here, and Randy and Alex have been going over this extensively, but basically just mirroring a little bit if we're interested in the epitopes. In this case, we're gonna put any. For the organism, we're gonna start typing our SARS-CoV-2, and IEDB is smart enough that if you start typing something, it's gonna autofill for you. And then because the data that I show you were based on human, we're gonna select human as a host just to make a fair comparison. And because we haven't looked into that yet, let's look at what happened to the B-cell data particularly. And another point I wanted to point out, and this has been already reiterated by other people before, the IEDB database contains both epitopes and also peptides that have been tested and been negative. So including the negative data is important, particularly if you want to do an ImmunomeBrowser analysis, for instance, because sometimes you don't know if a specific region is immunodominant because everything has been tested and only that is positive, or just because maybe the scientists, for whatever reason, have been only focusing on a specific region of the protein. Therefore, in that case, you're not going to see negative data all over the protein, and that can also suggest you there might be additional region on the protein that you could explore for your own science. So you're going to press the search button, and just to speed this up, I already did that for you. And we have several B-cell epitopes, and basically by clicking on the antigen tabs and clicking over here to actually check on the ImmunomeBrowser, and I'm going to just show that to you here for convenience. This is the type of results that you observe. So the data are showing in two different ways. The first one is the response frequency, where you can actually look at how many positive response over the negative. And also you do have an interval of confidence with the lower and upper bound. And the cursor over here can show you for each point of your amino acid sequence, you actually have a different type of response. There is another way to look at this data, which is instead based on the assay counts, and we have been discussing this already. And for instance, here you do see that there is a peak, and that is corresponding to the RBD region of SARS-CoV-2 spike protein, which we know is incredibly important for antibody response, for neutralizing antibody, and several different assays can be used to actually detect the response, which is also kind of the reason why we do see this peak over here. So all the data that you're visualizing, you are always able to download them in the favorite format that you like. You can also download the assay counts and everything that you visualize. And the other thing, if you're interested in looking at the actual epitopes, where they map and what is the sequence of identity, you can still do that. What do we mean with sequence of identity? as many people mentioned, the epitopes might not be exactly the same, or they might be related to a slightly different sequence. So the ImmunoBrowser actually mapped all the epitopes to a specific sequence, SARS-CoV-2 reference sequence in this case for spike, which IEDB conveniently show you over here. So you always know what this is related to. If you're interested in mapping these on a different sequence, or if you have your own epitope query, then you can always do your own ImmunoBrowser analysis by using the standalone tools. So this is what we see for the B cells. And as I was mentioning to you, we do have a peak of response in the RBD region, and we do have other regions that are actually recognized. So what happened when we look at the T cells? So there are two ways you can do this. You can either go back to your original query and basically down select the B cells, select the T cells, and then specify the class restriction. If you want to look at CD4, we're going to go for class 2. If you want to go for CD8, you can go to class 1. As an alternative, you can go to your original query and basically change the filter option over here in the left panel. So if we look at the CD4 T cells to begin with, and I'm gonna show you again the query that has been already pre-made. And I don't know how you work usually. I'm usually opening 300,000 tabs. That's the way I work. But luckily, IEDB always remember me what type of filters I apply, so I can always check what type of search I've been performing. And here, for instance, it tells me that I'm looking at T cells, and particularly CD4 T cells, because that's the MHC restriction over here. So if we click into the ImmunoBrowser, and I'm opening this for your convenience over there, you can see that the picture has a little bit changed with respect to what we saw. You do have way more immunodominant single region. It doesn't look as we observe for the B cells. And actually, the picture is very similar to what we've shown before. And just as a reminder, like over here, we have the RBD domain. And once again, you have a little bit of recognition of the RBD from the CD4 T cells, but it's still a poorly recognized region. So what happened if we look at the same query on the CD8? And now once again, here it helps me to basically show you I perform the same query, but in this case, I select MHC restriction class 1. Here are our results. The number of epitopes, of course, change. We are gonna look into the antigen tab once again and click on the spike glycoprotein. So here are the results, and similar to what I show you, there are a lot of peak of response. CD8 response kind of like recognize all over the spike protein, even the RBD domain, which is located over here. So this is great, and IEDB has been a powerful tool to actually try to collect all the epitope information derived by different experimentalists all over the world. One point that have emerged, and particularly critical in the context of SARS-CoV-2, is to understand how are the variants in this picture. And basically the question is, if I have an epitope or an immunodominant epitope, is that related to a specific mutation in one of the upcoming variants? So the IEDB has been trying to basically help the experimentals or whoever is using the IEDB to actually answer to this question. And basically, and this is specifically only for SARS-CoV-2 spike protein, they have been partnered up to actually implement the NCBI query that has been curated by the active trace and allowed you to select the different SARS-CoV-2 variants. So you can do that over here. You can see that there are the sequence are in the Pango lineages. You have several sub-lineages of the Delta and so forth. So for the purpose of just this tutorial, I'm gonna focus on the Delta variant. That is because, as we know, is the most frequent. And basically, let's go back for a moment, looking first at the CD8 T cell response. What you can see is that indeed there are mutations in regions that are considered immunodominant. However, Delta mutations are not spread all over the protein, and specifically, you do still have immunodominant region for the CD8 T cells that are still conserved. So this is great news. And it's kind of like in line with what I've been showing on a different paper this year, where we look at the general T-cell activity, and we found no difference at the population level across the different variant studies. What happened if we do the same for the CD4? And I'm gonna do exactly the same. I'm gonna go back and select once again here the variant I'm interested in, and the mutation pop up again. And as you can see, there are these two mutations that are impacting some region, but many of the other mutations that we found are not necessarily coinciding with regions that are immunodominantly recognized by the CD4 T cells. What happened with the B cells? And I'm gonna do that again for the B cells ImmunomeBrowser. So I'm gonna just select these again for convenience. And now let's look at these again. So some mutations are not really affecting immunodominant region recognized by B cell response, but there are these two mutations located in the RBD that are actually critical. And as many of you might know, these two mutations are the one that makes the virus more concerning, because locating in the RBD domain, these two mutations confers high capability on the Delta variant to actually infect. And also they are located in region recognized by neutralizing antibodies, which kind of concerns in terms of reduction of B-cell activity, which has been also shown by different studies. So with that one, I would like to finish this tutorial, and I will be happy to take any further question later. And thank you very much for your attention.